Assalamu alaikum dear student. Uh, this is the part 2 of theory class. We will cover chemical bases of the inheritance, and I will divide the material in 2 lectures. Okay, you can see a diagram on the right side of the slide. This is the skeleton of the Neanderthal, a species similar to human being, and probably this is said that it was a species from where the human evolved. And some people, uh, some scientists, they believe that the Neanderthal actually were replaced by the human being, and during certain time on the globe they lived together. So this beautiful structure is now present in the museum, and that was first named as Iceman after it was discovered in the Tyrolean Alps area, that is the between Austria and Italy. Just under the snow, it was discovered, so that's why it was named Iceman. So based on the radiocarbon, that is one of the technique for dating back the uh, existing existence of any species on the globe. Uh, this skeleton was 5,000 years old, and basically this Iceman was Neolithic hunter. So Neanderthal, they were also considered as a wonderful hunter. So DNA of Neanderthal or this skeleton was uh, extracted and sequenced. But more recently, an ancient DNA from the bone of Neanderthals was also extracted, that is at least 30,000 years old. So we can conclude from this information that DNA is the most stable material. Here is the website ENSEMBL, that is the European Molecular Biology Lab, and they have sequenced the Neanderthal genome, and you can browse this website to get more information regarding the Neanderthal genome. For anything that is to be genetic material, there must be some specific features that should make it unique to become a genetic material. It must be highly diversified, should have tremendous diversity, but the language should be same. It should be written in same or only one language. So three important characteristics for anything to become genetic material. It must contain pretty much complex information. That means it could be encoding for the information, and it must replicate faithful with highest level of the accuracy. And then whatsoever is this material, it must encode the phenotype. So it should result into the phenotype. Here is the history of the study of the DNA as a genetic material, and it's way back the discovery of the nucleus in 1833 by the Brown, and then the most recent one, the Waster and Crick uh, model of the DNA, structure of the DNA. Then the Frankel, Conrad, and Singer in 1956, after 3 years of the discovery of the DNA model or the structure of the DNA, they proposed RNA as a genetic material. You can go through the names of these scientists and what they discovered. Well, DNA basically, I have just mentioned some properties for anything, for any substance to become the genetic material. Fortunately, DNA does have all these characters, and this is basically composed of three uh, molecules forming a one single molecule, phosphate group, then the sugar, and the nitrogenous bases. Now, regarding the DNA, we know that it contains A, T, C, and G. There are four nucleotide bases. There is a Chargaff's rule, and according to that rule, we know that A is going to bind T, G is going to bind to C, but in any species, the ratio between AT and GC, it would be 1. And here in this table, you can see in the last column, the ratio is almost 1 for all the species. That includes microbes, E. coli, that is the E. coli, yeast, sea urchin, rat, and the most complicated species, the human being. So how DNA is a source of genetic material? An experiment shown over here. So there were two strains, S, that was the virulent, and R, that was the non-virulent. And when they injected the virulent strain to the mouse was killed. And when they injected the non-virulent strain, the mouse survived. Now what did they do in the next experiment? They boiled the virulent strain, so it was killed, and when it was injected to the mouse sustained and remained healthy, did not die. So in another experiment, they extracted or they used the non-virulent strain and mixed dead S strain, that was the virulent cells, with the living non-virulent strain. And as the S strain was dead and it was mixed with the live non-virulent strain, so the expected result should be the survival of the mouse. But the mouse in fact died. So that indicated must be some material that survives even after the death of the strain and resulted into the disease or the death in the mouse. And that material seems to be the DNA. Similar experiment was conducted by Oswald Avery, and he concluded the same. Then in that experiment they try to explain whether it's the DNA part which is responsible for the effects or whether it's the protein part. The result of this experiment proved DNA as a genetic material. Well, so once it was established that DNA is basically the genetic material, Then the structure of the DNA, that was the challenge for the scientists. A lady named William Ashbury, she actually first time conducted experiment uh, using crystallography or X-ray diffraction technique and transferred that image to the uh, film, but unfortunately she could not drive a structure out of that. Watson and Crick, they gathered that information, they studied those papers, those results, and they eventually gave the uh, structure of the DNA. Well, could RNA be as a genetic material? As I told you in the history, yes, it could be. So viruses basically they utilized RNA, not the DNA, in place of as a genetic material. So they have a bit different chemical composition of the genetic material, and that was first uh, reported by Frankel, Conrad, and Biesinger, 1956. Well, whether the RNA is a genetic material or not, the studies were also conducted for that, just like the DNA. So there are two viruses, TMV, that is tobacco mosaic virus, and HR virus. And what did they do? They degraded TME protein. The protein was removed from both of them, and they used the TMV RNA and HR protein for the folding of that RNA, and the result was the disease. So that indicated basically it's not the protein component, rather the RNA component that is responsible for the disease. Well, now the structure of the DNA, there are at least three levels of the structure of the DNA. And what are those three levels? We will describe in these slides. So primary structure, there is the phosphodiester linkages. Uh, then it's a macromolecule and it's a polymer basically. So polymer are those where there is a chains of molecule attached to one another. That's called the polymer in chemistry. So its unit is basically nucleotides, and these nucleotides they join together and form a polymer. Each one of the structure is basically composed of three parts. I previously mentioned the ribose and deoxyribose, or the ribose sugar, that is basically deoxyribose. Here we can see the structure of the sugar ribose. You can see at the it have five carbons. One, I'm showing with the green circles. 1, 2, 3, 4, and 5. Similarly, with the deoxyribose, again there are 5 carbons. 1, 2, 3, 4, and 5. Now, where is the difference? The difference you can see in the red circle. On the carbon number 2, there is hydroxyl group attached with the carbon in ribose sugar, while in the deoxyribose sugar, there is the hydrogen attached. So as the oxygen is removed, that's why it is called deoxy, without oxygen, and it's called the deoxyribose. That is primarily the component of the DNA. Then the sorry, second part is the nitrogenous base. So here you can see purine and pyrimidine. These are basic structure. The red rectangle you can see, and you can remember the positions of the each carbon 1 to 9 in case of purine. So in this basic structure, if on the position number 6 an amino group is attached, that becomes the adenine. And if on the position number 6 is the oxygen and the amino group is attached on the position number 2, it becomes the guanine. Similarly, you can see the pyramidal structure, that is the basic structure, and it has 6 carbon positions. Now, in case of cytosine C, you see the amino group is attached on the carbon number 4 and oxygen on the carbon number 2. Now, thymine is different to cytosine because both of the position, carbon number 4 and carbon number 2, they are attached with the oxygen group. And similarly, the oxygen is attached on the same position in uracil. And there is only one difference, and that on the carbon number 5 in thiamine, CH3 or methyl group is present, which is absent in case of uracil. And probably this is the reason that it is not as much stable as it should be, because of the absence of this methyl group, and the overall stability of the RNA is not as strong as that of the DNA. Now, phosphate group you can see, so there are 3 oxygen which are naked in the phosphate group. Well, based on this basic structure, we come to the structure of the each of the nucleotide. So AMP, GMP, TMP, and CMP. So that's called the deoxyadenosine 5' monophosphate. Monophosphate means there is only one phosphate group attached with the sugar. And deoxy because the sugar is used in this basically without the oxygen. So here you can see, if you remember the positions, this should be number 1, and this where the phosphate is attached, this should be number 5. So among all these 4 structures, position number 1 is attached with the nucleotide base, and position number 5 is attached with the phosphate group, and that's true for all of the structures. Now the only difference among these structures you can see in the red rectangle is the nucleotide base. If the nucleotide base is changed, the nucleotide would be changed. Rest of the structure is the same. Okay, the rest of the part of the lecture, or this part 2, will be covered in the next lecture. Thank you very much.